Welcome back to the next part here. We had left off model or lecture 5 of the membranes. This is a cartoon showing an early model of NSF snaps and snares in Golgi transport, okay, in between the stacks. So let's start with a transport vesicle, and this is shown here, okay, transport vesicle. Okay. Transport vesicles contain cargo inside, okay, cargo, and they have a protein, a snare protein called V-snare, vesicle snare. And if a Golgi membrane stack is the target, here's the target or destination has a T-snare. Okay, T-snare. And it turns out that the T-snare and V-snare interact through a protein here that's fatty acid modified that then recruits alpha SNAP and then assembly of the fusion complex, and that's NSF. There they are together. Energy is used, and this protein here, which is a RAB protein, they don't say it, but they should. That's what I study, RABs. Okay, RAB is a GTP case. And what happens is that the membrane fuses and the cargo is released. And then all these snaps and NSF, they're free to be recycled for another round. Okay, so it's a cycle. You need ATP and you need GTP for the RABs and acetyl-CoA, I forgot which enzyme that was. This is not the whole machinery at all, just shows you some of the machinery. Okay, we were focusing on the SNAPs and the NSF. NSF is the NEM sensitive protein. Okay, so apparently if you knock that activity out, then you're not going to get this process to occur. And so finding the NSF snaps and snares, so remember he could purify these vesicles and found the V snare. The stacks had the T snare, and he found, of course, the NEM allowed him to find NSF, which then, through the protein interactions, he found snaps. So he found vesicle-associated snares and target membrane, and those two find each other. So how is it that a vesicle made in one organelle interacts and docks with the right place? The snares determine then that interaction. So everybody's organelles have snares. So again, just down the road, efficiency of your snares coming together can be measured as well. And is that issue with the disease? If they fuse too quick or don't fuse enough. And then eventually the V and the T with the other molecules, NSF, snaps, they call it the membrane fusion complex that then causes the membrane to fuse with other material, other proteins, and delivers the cargo. Rothman's snare hypothesis. So they draw it, see it looks like a V snare, and see they drew the T snare together. Okay. So endocytosis has V snares that come into the endosomes, and these V snares don't recognize than, say, the T snares of the cis, right? They only recognize their specific partner. Here's V snares from the trans-Golgi network finding the plasma membrane. Okay, here they are. Here's movement from the ER. The vesicle snares recognize the cis. Okay, and so obviously retrograde, there's got to be snares as well, right? And forward and reverse in the Golgi, right? Forward and reverse, got to be different snares. So this was the original hypothesis. Incorrect V snares is discouraged and hopefully doesn't occur much. Depends on how efficient your cell is. So let's get a little bit more into NSF. So they found that these snares, when they bound each other, and they're not showing other proteins, and they're showing the T and the V snares here, red and blue. So here they are. Okay, they bind together. The docking actually requires a molecular twisting, and that brings the two membranes together. So energy is required for their fusion. So they're two separate Twizzlers that interact, and then they twist them even more. So the twisted Twizzlers, the snares, are here. Okay, so the result is we have a fusion. The cargo is delivered, but now we have twisted snares. And if we don't untwist them, then you can't grab another one. So they found adapter proteins. Here's our NSF and ATP. All of these bind these twisted Twizzlers and untwist them. Snare disassociation. Okay, energy's lost. And now these are free, okay, to dock again. When Rothman and Ross did NEM, it bound to the NSF, and that didn't occur. So you got one round, and you had twisted snares, and they never would untwist because NSF was inactivated. So it was inactivated, and NEM will bind to it and shut it down. So it, again, NSF with adapters allow them to bind to the twisted snares and untwist them. If you don't do that, vesicles stop. And so vesicles will then be around, and then Rothman was able to purify them because they couldn't dock because of the NEM. This just shows a little more. SNAPs are also involved in the targeting. Here's a SNAP 25. BOS, BET, syntaxins are all involved in the twisting and putting them together. Okay. VAMP is actually another name for a V snare. There are 4 snare families: syntaxin, BOS, BET, and VAMP, which are snares. And there's 4 twisted molecules, a 4-strand interaction that allows them to twist and the membranes fuse. So here's the different vesicles. They have a coat. Okay, that coat uncoats. See, notice right here, the coat's gone. So if this is ER, then that's COP2 and it's gone. And then here the snares twisting. And if NSF doesn't untwist them, then you've blocked vesicle fusion from now on. And here they are. If this vesicle binds to its cargo right compartment, then this will go there, this will go there. Notice the colors are different, showing the destinations are different for the docking and the fusion. The twisting, the water is excluded, and they call that a snare pin, the twisted snare pin structure. They form a stalk, and the membranes become so close that the lipids find each other, and you have a hemifusion and a complete fusion, and there's the cargo coming through. Okay. And there's more proteins, but we're just talking this. So that's a snare pin. So turns out that hemagglutinin in viruses, look at their HA. It looks like snares. Okay, so it will come in and function, trick the donors to act like it's a snare and twist, and the viral membrane will fuse, and the virus then is released. So viruses have an HA protein that looks like our snares, and that's why viruses spread so well, is because the machinery allows them to fuse into cells. And once it's inside a cell, it's hard for the immune system to get it. And so it's interesting that our viruses of humans actually have snare-like proteins. And a snare pin is formed, etc. So the protein I study is known as RABs. RABs regulate all this targeting and fusion. It was isolated from a genetic library, a rat brain library. They bind GTP and when to be active, and when they're not active, they're in the GDP found structure. They are found concentrated in transport vesicles. So finding them, they probably purify the vesicles, and they found these GTPases, these RABs here. We have different RABs for different transport pathways, and various RABs have their own place where they hang out and do their business, different organelles. Large amount of proteins that interact with RABs, those are called RAB effectors. And so they don't have just one function. Humans have 64 different RABs, not all of them in every cell. Okay. If you're a neurobiologist, RAB3A is the RAB involved in exocytosis of neurotransmitters. So when they found those secretory granules in fusion, they found RAB3A. They are thought to be the molecular switches that turn on and facilitate membrane trafficking, and they thought to modulate the snares, binding and twisting and bringing the membranes together. So here is the RAB GTPase complexed cycle. So here's our T and V snare. So let's start it. Here's a vesicle forming, and here's a RAB that's bound by membrane protein, so it's stuck there, and it's right next to the V snare. The vesicle is released, and the RAB is in the GTP form. It's not showing the coat, right? There's coats, and then the coat is removed. Here's the RAB. And it turns out that when the snares find each other, then there's NSF and the snaps, there's going to be the fusion. RAB is sitting on the vesicle, and there's a protein known as GAP, GTPase activating protein, and it puts RAB in the GDP form, which is now RAB is off. So this is the off RAB, GDP, okay, off. RAB GTP is on, okay, on and off. And there is a protein called GDI. It's an inhibitor. It binds. It allows the RAB to leave the target membrane, be recruited back to the donor. The GDI is released. The RAB is delivered. And there's a GDF that displaces the GDP and adds the GTP exchange factor, GEF. Okay, and now the RAB is active again, ready for process. So RABs must switch between GTP and GDP. Part of my work is figuring out where they're at and what they're doing. We actually have a mutant that's stuck in GDP form, and it causes changes in vesicle movement and balance in the cell. Okay, all RABs have a cysteine at the end, right here, and that's where they get germinylation. It's a fatty acid. And they're become isoprenylated by two geranyl fatty acid chains. So there's an enzyme that decorates the RAB and puts them in the membrane. And here's different RABs, different places in the cell. So RABs are the busy beavers, and they work specifically in certain areas. This is just a cartoon showing some. So not only are the snares involved in causing the right fusions, but RABs need to be in the right location. So RABs can be cytosolic or membrane associated. They jump on and off the membranes. Some RABs can be with other RABs. So here's RAB5 for endo. RAB4 is exo. Okay. RAB11 is in the late, and it moves vesicles out, or RAB4 can move vesicles out. So it's a super kind of a later endosomal complex here. And so RABs are recycling between donor and acceptors. And I believe we've seen that. Here's all the RABs. Not you have to memorize any of this, but I've done a lot of work in RABs in my organism. So while Rothman and his group were isolating the snares, the NSF, the RABs, the machinery, they were finding that there were other proteins involved. And so one of those is found by Sheckman's group in yeast. It's called SEC1. And here's the V snare, the RAB, they're not showing the coat, but the coat's formed and then removed. And now you have T and V snares. So what they found was that this SEC1 protein is like a hat that the T snare wears. And so that hat stays on until the right V snare comes along and it is removed. So you tip your hat to the V snares, and now the RAB with others regulates, and then you get a fusion here, and it showed the fusion event occurring. So part of the regulation is you cap the T snares to make sure they only are available for binding to the correct V snares, and SEC1 does that. Okay. So if SNEC1 is stuck, then you would never get a fusion. Or if SEC1 never binds, then you might get fusion of the wrong vesicles to the wrong compartment. SEC1 is critical here. So here's our VAMPs or V snares. Here's our T snares, also known as syntaxins. Here's the SEC1 hat. Here's the RAB protein. Here the snares are trying to find each other. Here's SNAP25 is regulated, put in. Here's our hat. The hat is removed. Then you have the NSF and others are coming in, the snaps, you're twisting. Okay, so the twisting, and then the hat's removed, and then the core complex, the junction or fusion, the snare pin is forming right here. Okay. So they thought the vesicles can be tethered together, and that allows the vesicles to be in the right place as well. So there's the concept that there are other proteins that interact with the vesicles and kind of keep them towards between the boat and the dock there to allow the fusion to occur. So as we see with vesicle fusion, it turns out, and we always wondered, how are these Golgi stacks attached together? Turns out they are attached by snare-like molecules. Here's Golgi stack A and B, and it turns out there's a protein that's very large called gigantin, and GM130, that's its molecular weight, and P115 as well. And they show that P115 tethers the gigantin and the GM30 together in the process of GRASP 38, so GRASP and P. And so what happens is the matrix are bound together by the binding of the 115 and the GRASP molecule, and now the two stacks are fused together. Well, they're not fused, but they're held close together, so there is some, that's how we can see a stack here. So there are proteins that mimic the V and T snares to bring the matrix together. So you would reverse this process probably in mitosis to break the Golgi back up. So you have a regulation of the stacks here. And so the P15 in the Golgi is direct row with tether and snare pin. Here's P15, which has a friend, Rab1, and Rab1 puts the gigantin and 130 together, and here they are interacting with some other proteins, and it's thought to seal them together. So snare interactions are important, but there are other proteins that make sure that they're connected as well. So here's a model showing the whole transport process here. Here's a donor compartment. Here's the receptor, cargo. Here's a coat forming. Here's our friend the Rab. There's the snares. Here's our sec protein, which is covering the T snare. Okay, here it is. Then the Rab with an adapter, Rab effector, tethers it. Notice the snares are finding each other. The sec1 is gone, the hat's released. And then the fusion occurs, and then the Rab is converted to the GDP form. You've twisted, and then NSF comes along and untwists the Twizzlers, and now they're ready for this process again. And then sec1 would bind, come back in here and bind. And the Rab goes back, okay, is recruited back here. So that's the cyclic pathway involved in vesicle trafficking. And the trafficking gets really complex in cells that have two different fronts. Our epithelium cells have a side facing the environment, the lungs, the blood vessels outside called apical, and those sides that are found to the attachment here is said to be basolateral. And there's tight junctions between these cells and our epithelial here. So now in these cells, they're very advanced because there has to be information going to the right location. So there has to be extra zip codes here, just showing the apical versus the basolamin, the basolateral, and showing the different proteins there. So they have apical and basolateral endosomes, and they're showing that they're endosomes that are basolateral in green or apical in red. So there's more information, and then here's a fusion where the late endosome where both vesicles meet. Doesn't that look like a little rabbit or an upside down duck? Yeah, Here's the little rabbit here. See his little ears? Okay, and then this can interact with lysosomes or become a lysosome. So there has to be material that makes its way out to either one of these sides, okay? And then these tight junctions keep the membranes separate here, these tight junctions. So epithelial cells are very unique cells that way. So it shows that there are early endosomes, basolateral and apical endosomes as well, showing the interaction and showing some of the pathway. There is transcytosis where there is material that can flow through, okay, in either direction, but they require specific transport and cargo. Here's glucose, comes in, sodium comes in, and they have a glucose sodium symport. That means they both go in the same direction. Here's symport. Antiport would be they would go in opposite directions. Symport, one direction. So you end up with sodium and glucose that then you want to get inside our blood or lymph here to feed our cells. There's glucose from the intestine. And then the potassium can flow out, and that drives, sodium out, and that potassium can come in, or it drives other molecules in as well. And that's facilitated diffusion. There's an inhibitor of this ATPase, Wabane, and if you add Wabane here, no effect. If you add Wabane to this side, it inhibits this whole process, and that will slow down glucose coming in. Just poison, Wabane, because you don't start getting glucose in. You need glucose for Krebs cycle and whatnot. And here's transcytosis, where a material goes in, fuses, and then you hear the two vesicles fusing. Here's transcytosis. So IgG in blood can be picked up and moves through, and in mother's milk released, so antibodies to the baby getting fed through transcytosis. Polarized means having two distinct regions here. Here's IgG transposed in neonatal blood, so you're getting antibodies over here to protect the baby, and that's called transcytosis.